So, thank you all for joining today. This is almost towards the end. This is week 7 of the NPTEL-PMRF scheme sessions on the course Tissue Engineering from IIT Madras. So, this week we have studied about the host integration and immune responses, and what are the factors, what are the key players, the key cellular components, as well as what are the properties that needs to be taken into account to modulate the host integration and immune responses, so that we can have a successful graft integration or tissue engineering application that we have studied. Then we have also studied about the bioethics and what are the dilemmas in tissue engineering, as well as various concepts of bioethics that we have studied. We have also extensively studied, particularly on the tissue engineering applications of the what are the various tissue engineering concepts, as well as what are the recent advances we have studied about some commercial aspects also. What are the commercially available tissue engineering materials that are available? We have also studied about, right? So, I wanted to start today with this question. A patient visits with chronic wound. He reports a previous medical history of alginate allergy. What among the following dressing materials would you suggest? Options are given as Yetane dressing, Comfil Plus, Durelon, and Tagagin. Question goes as patient visits with chronic wound, okay, and he reports a previous medical history of alginate allergy. What among the following dressing materials would you suggest? Responses. So more than one option is correct for this actually. That's why I put this question. So the correct option is, alright, let me correct. So this is not suggested. I'm sorry, okay. So these two of these are not suggested. Durelon and Tagagin would be suggested, okay. I'm extremely sorry. Why and these two, Biatin and Comfil Plus, we are not going to suggest. Comfil Plus is also alginate based, right? So the question is, he has a previous history of alginate based, alginate allergy. Yeah, Comfil is also alginate based, and Biatin also alginate based material. Okay, so there's two options. So Durelon and Tagagin we will be suggesting not these two. Biatin dressing, Biatin is also an alginate based material for wound disease, a commercial material along with the Comfil Plus. Okay, so what were the options that was shown in the lecture? If the Biatin, I want to add it, the Biatin dressing also. Okay, so Biatin dressing and Comfil Plus both are alginate based. The patient has the alginate allergy, so we will not get using it. Okay. So for this question, this two would be suggested. So basically, what I wanted to talk about is this kind of commercial dressing and what are the materials that are used, what kind of biomaterials that are used is very important, because these questions are really important and to study very deeply. Okay, so that lecture slide with the table, do read very carefully and don't make this kind of mistakes that I have made. So it is a bit confusing sometimes, because there are lots of options and lots of material that was given. So don't get confused with this kind of options. Okay. Alright. Moving on to the next question is, which of these molecules plays a role in homeostasis during wound healing? Option goes as epinephrine, fibrinogen, collagen, and elastin. Which of these molecules plays a role in homeostasis during wound healing? during the homeostasis phase, which of these molecules play a key role? The correct option is epinephrine. Yes. Collagen and the other these options comes a little bit later. So when after the foreign reaction and these steps are coming in, so that time this molecules fibrinogen, collagen, elastin, these molecules come into picture. But when the homeostasis phase, as soon as the wound is happening, at that phase, this epinephrine molecule plays a key role during the wound healing process. Okay, so this is a very primitive stage. For the homeostasis phase, in this state, the epinephrine will be the key mediator. Alright. Okay. Next question is, which of these factors affects cell migration through skin grafts? Select all that. Option are migration rate of specific cells in the skin tissue, availability of gases and nutrients, thickness of the graft, and all of that. Which of these factors affects cell migration through skin grafts? Select all that. Yes. All these factors, migration rate, specific cell types, as well as the availability of nutrients and gases, as well as thickness of the material. All these factors act in a combination for this cell migration through skin grafts. Another question is, select components from the list below that will be useful for construction of a tissue engineering skin graft. Select all that. Sites, metablasts cells, polyethylene glycol, melanocytes, vascular endothelial factors, and F collagens. All these options select the components which will be useful for the construction of tissue skin graft. We need a material. We need a material from which we will be making this skin graft. Right? That's why we're using a polyethylene glycol. And then other factors, this keratinocyte, fibroblasts, melanocytes, vascular factors, as well as this. All of the factors would be equally important for a successful construction of a tissue engineering skin graft. Right? Practical application. All these components would be needed. Sometimes this option like why PEG? PEG is we need a material to fabricate a scaffold. PEG would be for lowering the immunogenic responses of the scaffold. As a like useful concept of tissue engineering skin graft, we need some material that will be used for particularly for a material that will give lesser immune reaction when it is impacted in the site of application. Right? PEG is that kind of molecule that will give lower immune response. We will be talking about it in detail. Okay. So this why using PEG is because of this foreign body response that we have studied. Okay. I want to talk about it a little bit because all the biomaterial tissue engineered grafts that we have talked about throughout the lectures, previous lectures, all of them come to this whenever these implants are taken to the in vivo stage, the in vivo application inside body. This APR or foreign body response happens. Right? So, what is the first few stages that first we can see the protein adsorption. So, all different protein components like C5 complement system components, C5, C3, as well as albumin, globulin, and fibrinogen kind of proteins, they come and adsorb on the material surface, biomaterial surface. That is the implant. Then the recruitment of innate immune cells, that is the neutrophil, monocytes, and macrophages, all this kind of innate immune cells come and home to that region, and then this high level of protein as well as this and the chemoattractant gradient that is created because of this protein accumulation to the material surface, there is a homing of all those immune cells. Especially at first comes the neutrophil, and using this neutrophils and secreted molecules of neutrophils, other immune cells are homed in that region. Okay. So as this immune cells are gathering in that region, for example, monocytes also come, and from monocytes the macrophages and the macrophages are then fuse and form the foreign body giant cells, or MPGCs, multinucleated foreign body giant cells. So this kind of large multinucleated cells are also formed and engulf the bone material, at least tries to engulf the bone material, shielding the other cells access to that material. As well as there are various kind of proteinases that are released from these MPGCs, which ultimately results in sometimes degradation of the materials, because one of the materials we are using is either carbon or protein based. Right? So when these proteinases are being released, so that results in the degradation of the material before it can actually function. Right? So that is called the graft failure that we have because of this MPGCs or foreign body reaction. So to minimize this, traditionally one can think of one strategy, that is using some kind of material that will not even elicit this kind of foreign body reaction, an inert material that will not give rise to this APR. So some materials like which are functional group, enriched functional group of COOH or OH or enriched to discharge functional groups. There are some deuterionic elements, deuterionic polymers. This kind of polymers sometimes are used that reduces the protein absorption. If there is no protein absorption or there chemoattractant gradient, there will be lesser immune reaction. So that is one classical mechanism, withdrawing from the fact that there will be a immune cells migration or their immune reaction. A chemically inert polymer method that is used, and so that the APRs are minimized. That is one. Another mechanism is using this APR polybody reaction to our purpose of interest. Why? Because the macrophages that are created are somewhat specialized according to the tissue environment that is in the picture. So there are two different kinds of macrophages that come into the picture. So when the neutrophil, if I say it in a good way, divides the monocytes there, it divides into, or it forms two different kinds of macrophages. One is pro-inflammatory macrophages. Where the pro-inflammatory macrophages are only functional in the it functions for the destruction of the material or destruction of the foreign body that is present. Foreign body means whatever the antigens or the material that we have. That is the function of the pro-inflammatory macrophages. We call it the M1 macrophages. And there are another class of macrophages that is created from the M1 macrophages. That is the M2 macrophages. That is the pro-regenerative macrophages. Okay. So this M1 and M2 macrophages, sometimes in combination or in sequence, or M2 macrophages alone, can function to help the tissue injury activation or care wound healing responses. So our target would be, for a successful tissue injury application, our target would be to have maximum number of M2 macrophages in the population of the cells in the API. That is the use of this immune response for the tissue healing purposes. So one using inert material, withdrawing the immune response. Another is using this immune response for helping the tissue healing application. So you can see here this immunomodulatory biomaterials, this kind of specialized biomaterials that helps in the enrichment of this M2 macrophages in the engineered area. So it happens by various mechanisms. For example, there are various stimuli molecules like IL-4, IL-10 or miRNAs. These stimuli molecules can be delivered through the graft that we are inventing. So we can have synthesized a kind of graft that has this kind of stimulatory molecules embedded in it, and it is being released as the engineered part. where we have placed the graph. So what it will do, this kind of simulatory molecules will start enriching the into macrophages. That is one strategy we can have. Second strategy we can also have is mimicking efferocytosis. Efferocytosis is a mechanism where apoptosized neutrophils are engulfed by the macrophages. Now, neutrophils, when the apoptosis occurs, there is a phosphatidylserine enrichment. So, phosphatidylserine molecules are only mediator of only present when there is an apoptosis in the body. Okay, so when this neutrophils, apoptosized neutrophils are engulfed by the macrophages, so this phosphatidylserine molecules does not get the chance to tell the into macrophages or any kind of macrophages to engulf any kind of other materials. So it is blocking the macrophages receptors to further engulf any other materials. Okay. So it is just blocking mechanism of macrophages phagocytosis mechanism to save our graft. That is another mechanism. Third is sequential activation. First, activating the M1 macrophage, then using the M1 macrophage's released growth factors to further evolve to the M2 macrophages. Okay. For example, if we supply our graft with the interferon gamma like M1 macrophage proliferating substance, then as the M1 macrophage population is increasing, it will then again start supplying the M2 promoting cytokines or drugs like simvastatin or growth molecules like interleukin-4. So this kind of molecules when sequentially applied, so one M1 enriching molecule and M2 enriching molecule sequentially. After M1 is enriched, then the M2 will be triggered. Okay, so that way we are getting a larger population of M2. So our main target is M2 macrophage increase. M2 macrophage population needs to be increased. As well as there are various MSCs, that is mesenchymal stem cells, that is a cellular therapy. For cellular therapies, the MSCs are also tissue engineered, or there are various genetic material inserted to the MSCs that will release these kind of growth factors and stability. So using those specialized MSCs, along with the cell therapy, cell and biomaterial lab, we can have a successful immunomodulatory biomaterial for a successful tissue development. So, classically, withdrawing from immune response or using the immune response for a tissue engineering response, tissue engineering application, we can have a successful application on tissue engineering. So, this is the foreign body response and using the foreign body response for our purpose of tissue engineering. Okay, I just wanted to just clarify these points. Alright, so this is a true-false question that I asked. So the first option, each of proteins are an example of alarmins. It's a true statement of false. Each of proteins are an example of alarmins. Each of proteins. What are alarmins? Alarmins it activates what kind of cells? If here each of proteins are example of alarmins, if it is alarmin is not there, it is in the DAMP. DAMP is not DAMP, it's DAMP. What would be the options? What would be the correct answer? True or false? Alarmins, instead of alarmins, we write DAMP. It will be true or false? So DAMP, that is damage associated molecular patterns, it is also called alarmins. So alarmins and DAMPs are synonymous. So So when if there is an option that is say that DAMPs, HSPs or any other molecules are example of DAMPs, then it is the same. It is true. Okay. If there is no term or alarming in the question, don't get confused. DAMPs and alarmins are the same. Next one is alarmins are recognized by B leukocytes through pattern recognition receptors. Is it true? Alarmins are recognized by B leukocytes through pattern recognition receptors. Anyone else want to try? Spots. What kind of cells that recognizes alarmins? What kind of cell? So they will recognize as macrophages that are formed in that particular tissue, particular part. Before the APGCs are formed, this kind of alarmins trigger the formation of APGCs by the macrophages. Because the alarmins are synthesized and alarmins are secreted, using these alarmins through the PRRs, the macrophages try to engulf the whole mammalian. Now, therefore, it needs to be fused. The APGCs are formed, not the B leukocytes. So alarmins are those kind of molecules that are that tells the cells that before the APRs is formed, that the material is not the site is injured, and there is a material, a foreign material that has come. That is, we have implanted some biomaterial here, a foreign biomaterial. So for this message is recognized by the PRRs present on the bacterial receptor because of the macrophages. That is moved in the particular biomaterial. Right. So alarmins are recognized not by the B leukocytes. Different kind of cells in macrophages through PRRs. These alarmins are recognized. Next, polymorphonuclear leukocyte releases the additive substances such as that can result in cell destruction. The next is foreign body giant cells secrete protons, enzymes, and reactive oxygen species in an atom to phagocytose implantable biomaterial. Is it okay? True or false? Foreign body giant cells secrete protons, enzymes, and reactive oxygen species in an atom to phagocytose implantable biomaterial. It is also true. Okay. Alright, next question. Identify the option that describes the phases of normal wound healing process in the correct order. homeostasis, proliferating, remodeling, and inflammation. With these four options, if you organize, what is the event order? What happens during the healing process? Why you are saying inflammatory, proliferative, remodeling, homeostasis? No. When the wound healing process occurs, what are the events of it? For example, I have had a cut in this region. What is the first thing that is going to happen? The wounded cells will be releasing certain chemicals or certain growth factors or something. This crude way. Our wounded cells will be releasing certain chemical elements. So that event is called the homeostasis. Okay. Then, according to these chemical gradients, there will be an inflammation because there will be some bacteria, not the chemical gradients, because there will be some bacterial infection. There is some other material will be there. So that will be first. Second, the inflammation. First, the homeostasis occurred, then the inflammation switch. Okay. So our immune cells have come and try to clear the region with all the microbes and all the pathogens that can. Alright. Then what will happen? Our cells, the M2 macrophage will transfer to the fibroblasts, and there are other cells coming in. So, as the homeostasis will occur, the inflammation. Whenever you have a wound, there will be at first the release of certain chemicals that is coming into the picture. Right? So there will be certain growth factors, there will be certain cellular entities that is coming into the wounded area. That will create the first homeostasis of the area. It will see the homeostasis. Okay. After attaining the stability, the inflammatory, that is the immune molecules that will come. Then the proliferation will occur. The proliferation of the cells, immune cells, as well as the host cells. And then the tissue remodeling. Okay. Think about the events. Events, what are happening? You have a cut. It needs to stabilize first. If the blood will clot first, then the injured cell will release certain growth factors, and there are certain, and depending upon those growth factors and those signaling elements, immune cells are coming in from the severe vesicles. Okay. Using those severe blood vessels, there are inflammatory immune cells are coming in and enriching the area. Okay. Then those cells are transforming. These ABRCs are transforming to various other kind of cells, or the ABRC bonding on these events are occurring. All the macrophages and macrophages population, the various other cells are coming into the picture in this. So these proliferative cells now. Yeah, homeostasis means this is the at first the site needs to be stabilized, right? That's why it needs to be first step. Then the last stage, there is the remodeling, tissue remodeling. That is first the preliminary kind of collagen and everything, the preliminary collagens and everything that has been secreted by the cells, that will then mature. That's why the last stage is the remodeling. So when the preliminary wound heals, the kind of cut that we have, if it is not completely which one? We are talking about the homeostasis. Right, homeostasis and hemostasis, whatever the option that is available. That is the first stage that will occur. Okay. The last stage would be the remodeling. Yeah. So the first stage needs to be stable. The stability needs to be attained for the wounded region. Then the inflammation, then proliferation, then remodeling. Okay. The last stage would be the remodeling. Okay. what are the recent advances in 3D skin models? So 3D skin models that we have, not only just for, as we have learned in the bioethics lectures, there is a constant need for these in vitro models, and we are trying to get away from the in vitro or animal models because of the obvious reasons that we have studied. So, what are the advances that has happened? So, we have studied about microfluidic based devices. We have also studied about 3D printing and all this. We have also studied about what is the importance of mechanical stimuli. So, using all those concepts from the previous classes, here I have given an example. This dynamic flow 3D in vitro skin models are made today. So, you can remember last class we have talked about a transfer insert where co-culture or where there is a separation between the culture container can be created. So, this kind of inserts are used, and using the dynamic fluid environment, we are creating a 3D in vitro model. This 3D in vitro models can be created. So, what advantage does it have? It can create using various layers of cells or various kinds of cells. This 3D in vitro skin models can be created. So, it would be like a more towards a practical whatever we have in the inquiry condition. Okay, those kind of in vivo like 3D in vitro skin models can also be created. Okay. So, this is the model 3D model that has given, and you can see there's various kind of cells, not only just one kind of cells, there are 2 or 3 different kind of cells that are cultured, and then sequentially. At first, the fibroblast as well as endothelial cells are cultured. When this formed vascularized cell there, then the keratinocytes are also there. See, our skin has various layers of cells. There are various epidermis, dermis, and hypodermis, and all those layers we have. So their skin cells are also different. So using those various different kind of cells, these multi-layered structures can be created, and these inlets and outlets can be used to provide a dynamic shear stress using the fluid. And using that device, more in vivo like 3D model can be created and used for other applications. One of the applications is drug screening application, drug delivery application. This kind of application can be for using this kind of 3D models. So that is a 3D printed one. Now we also have studied about the microfluidic based devices. So microfluidic devices are also using this dynamic fluid flow systems. This is a skin on a chip. So various layered skin cells are cultured, and then a dynamic fluid flow system is created, and then the whole system can be used. And you can see that there are an array of chips can be used, and there is multiple inlets and outlets. So what we can essentially achieve is we can have a study where multiple applications or multiple drugs or multiple tissue engineering applications can be performed in a single chip at the same time. Okay, so there will be any difference in the study parameters. Okay, so this kind of microfluidic devices are also being studied about. So another thing we have studied about is electrospinning. Okay. Now, handheld electrospinning has also been studied very recently. So what they have done is they have also used in situ cell, use it for in situ assembly. So they have the polymer solution with cells, and the whole thing is electrospun into the inlet side using a handheld device. The principle is same. The potential difference is created, but it contains the cells also. So you can see here in the same images this D. There are cells trapped into the nanofibers that are created using these handheld devices. Okay. So electrospinning in situ with cells for the welding application, that is also being done for the tissue of. Now, they have not stopped there. They have used a 3D printing like device for a thicker layer of mold elements, that is a deep mold heavy application. They have also done using this kind of device. This is a prototype of that device. You can see two syringes they have used with multiple materials. The fiber-based iron they have used as well as the cell source they have used. And using that, they have printed a layer of material on the wounded area, and they have given the skin model, 3D in vitro skin model. So not only just electrospinning, this 3D in vitro models can also be created using this 3D printer like devices. These are the handheld devices. Okay. Alright, the next question is what kind of immune responses relate to the following events? What are the events? The first is the killing of microorganisms due to low pH in the stomach. So what is this event? It's an innate event or the adaptive event? Next is entrapment of foreign microbes by mucosecretion. Is it innate or next is disruption of bacterial cell wall by lysosomes. Yes. Bacterial cell wall disruption by lysosomes. Is also the last one, antibody production by B lymphocytes against foreign agents. So what are the primary events that are occurring? The first introduction of the antigen in the system, that is taken into by innate system. Then the adaptive system get triggered and we get the response. Okay. So whatever this primary, whatever the primary events that may occur after the patient, whatever the application in our body, introduction in the body, this is all taken care of by the innate system. Okay. The rest will be to the adaptive system. So these are the questions I wanted to discuss today. So this is almost toward the last that I get more time for asking you. If you have any question for this 7 weeks we have crossed, if you have any questions or any topic that you want to discuss, again, identify with the lectures. Are there any topics? Exam preparation, we are expecting I cannot disclose the exam right. It's not policy. But what I can assure you, you will not be facing any kind of challenges while attempting the exam. It will be through the syllabus, from the syllabus, and especially from the slides that we got. What I can also tell you is focus on those concepts, basic concepts, as well as the information that are given, particular information for a particular scenario. For example, the last few lectures there was tables of what are the commercial implants, skin implants that are available. So those tables are important. So study about those tables. What is the kind of material? You don't have to remember all the components. Just remember what are the material that is used and why that kind of material is used. What kind of biomaterial is used? What is the strategy that is used? It is electrosmart, it is hydrogen, it is why it is used. What kind of material is used? Okay, focus on that. In the initial lectures, there was various concepts regarding the stem cells, the grafts, the various kind of grafts, this kind of concept, important concept regarding the tissue engineering, because we are studying tissue engineering. So this kind of concepts we need to study very, very well. Don't worry about the pattern, it will not be created cell migration to problems are given complex. It's not complex. What kind of questions, like what kind of problems you are looking for? Cell migration, even we have studied cell migration, how cell migration is happening, what kind of cell migration that is there, how to test the cell migration, what affects the cell migration. Concept of Brownian motion. So, see, Brownian motion concept is introduced, or the persistence time of Brownian motion. So, Brownian motion, when it was introduced, it was just to tell you that the cell's motion is random. Why the Brownian motion particularly was introduced is Brownian motion is a random motion of the body. Right? Cells migration can also be random. If there is no chemical magnet or any other influences there. Right? So to tell you that this Brownian motion concept was introduced. There is a migration and Brownian motion concept was because of that. Now when there is a signal or external signal, like electrical signal or any other chemical signal was there, then this their motion would be in a particular direction, either towards or away from the chemical or the external signal. So that's why this Brownian motion concept was. So like that, this cell migration, and you can follow this slides that we have prepared here, right? The slides that we have given, there are links, papers that we have used. Like today's lecture, follow the chemical reviews channel on the immunovulnerability parameters. What are the various strategies for the immunovulnerability? So there are various concepts of various kind of cells that are involved in immunovulnerability, and how to use that condition. This kind of papers are also going to be helpful for implementation. Okay, and we can get so I'll be focusing on the next weeks or 10 days, 20, 30 minutes. So there is time to be that was very so the hall tickets will be given after you have the registration done, and you will be intimated to you how to get the hall tickets and all the arrangements will be good. Registration is completed. As soon as the notification comes, you will get the intimation through email as well as the announcement. So I will not be able to disclose it right now because it is fixed. But it can be both. So this time I am not being able to disclose it. But yeah, it's assured it is not going to be a job. It will be a very easy paper. It's going to be it's okay. Just study about the concept and whatever the material that was provided, that is the slides that was covered, all the lectures. So we'll see. The pattern of the exam will be disclosed after the notification comes out. So we'll see. Any other question of the course where you might be really updated. So last, like the next week will be the last. So please write all the questions in the discussion forum so that I can get back to this question in the beginning. Okay. So, and try to gather, summarize all the part, see where you are facing it will be from the content only, lecture content only. The questions will be the lecture. There will be nothing outside the question. All the answers, all the questions also are there in the lectures. So you study with most attention. Now, another thing is there is a survey going in the board. I would encourage you to find this organizations that help to during the course time to write about your experience in the survey and weekly analysis with the results that you have gone through the lectures and what do you think could I do in the next period of during the sessions that would help you next sessions. Okay. Example problems, example problems needs to be prepared for. So, yeah, as well as the concepts and what are the examples that are given, both are very important. So concept you need to be clear about, then the examples you need to study about. So, as much clear you are in the concepts, you will be facing no difficulty in addressing the example questions or practical questions. So, do read the questions very attentively. Do not go to the decoration of the question. Try to understand what the question is asking. And accordingly, you answer. Do not get into the details of the question, what kind of extra details that are given. That might be the confusion. So don't get into that details, just try to understand what is the demand of the question and accordingly you answer. Right? So again, to just get a quick recap of the course content. Suggest this week along with the week's content. Just try to get a quick recap of the course content yourself and try to see like if any concept or anything you can find any different. To write about them like 1 to 2 days before, as we do 2 days, combine all those together with the slides so that I can address them again and we can clear it out. Okay. So yourself, do a quick recap and see where the problem areas are and we'll be addressing it in the last next one. Okay. Thank you for joining.